Zhengming, are you all set? Can I get going then?
I'm ready. I've got slides. Looks
great.
Yeah.
All right. Welcome everybody to the first educational seminar series of the International 100K Plus Cohorts Consortium. My name is Jeff Ginsberg. I'm a professor at Duke University, and I am the co-chair of the IHCC. It's really a pleasure for me to welcome Zhengming Chen as our first speaker in this series. Zhengming has been a pioneer in the area of biobanks. He is a professor of epidemiology and director of China programs at the Nuffield Department of Population Health at Oxford University. He is also an inaugural member of the executive committee of the IHCC and has been the impetus behind the China Kadoorie Biobank, which now holds information on over 500,000 individuals. And today he's going to talk to us about establishing and managing large population biobanks across geographically diverse areas. And we're really excited to hear Zhengming's wisdom on this topic. So the floor is yours. And I'll say one more thing is that we want this to be interactive. Please raise your hand or put questions in the chat for Zhengming, and either during the talk or afterwards, we will have a chance to reflect on your ideas. Zhengming, welcome.
Okay, thanks very much for the kind of introduction. So I'm very delighted and honored to be the first speaker for this seminar series. My thanks to the organizers for the opportunities. So the title is quite broad, general, but I think what I'm going to cover is mainly what we have gone through with the China Kadoorie Biobank, which is one of the largest blood-based prospect cohort study, which has been going on for over 15 years. I hope through the talk, it can sort of help promote scientific collaboration, not only the data sharing, but also sharing of the research methodology, but also inspire new studies in many low middle income countries, especially Africa, where there's a substantial unmet need. I think before I go through the kind of slides, I'd just like to spend several minutes and remind us some of the basic principles of the epidemiologic studies, especially prospect cohort studies, and some of the challenges, because that's actually quite important, will inform the study design in various ways in different countries. So first, I'd just like to see where we are now with our understanding of epidemiology. So many big causes of adult mortality are known, and thanks to the 20th century cohort study largely, like Framingham study, like British dot studies, American cancer studies. So this just shows some of the big causes, maybe in kind of a descending order in terms of the relevance for health: tobacco, alcohol, chronic infection, a wide range of them: HPV, HCV, HIV, and then blood pressure, lipids, adiposity, genes. This is still another major area with a lot of activities, and the diet and air. So through our understanding of these major causes of adult mortality, we see huge improvement over the last few decades in various ways: prevention, treatment, across many kind of countries, not only developed countries, but also in many developing countries such as China, India, and with a few exceptions like Russia. It's driven by particular historical events or episodes that are called binge drinking as well. So this is very encouraging, of course, with COVID-19, and we will see some slowdown or short reverse, but with vaccination, I'm quite confident, the trajectory, downward trajectory will continue, over the next decades or so. But despite our understanding of these major determinants of chronic disease, they do not seem to explain larger geographic variations between different countries, and also between different populations within particular countries. I just want to show this particular graph. This is a map of China. Two major cancers, esophageal cancer, nasopharynx cancer, it is known for many decades there's a large unexplained variations with some areas hotspot like central China, Henan, the incidence mortality is per 5-10-fold as high as neighboring countries for reasons we still don't understand. I mean, 40 years ago, the NIH put about $20 million into a huge collaborative intervention trial of a nutritional supplement, but it does not seem to review much. So there's still a lot of unknowns. So this is actually quite important, suggesting there's still many important environmental as well as the genetic causes await discovery. So that's quite important. And in a way, it reflects, the complexity of the chronic disease. It's nature, nurture, environment, genes. There are complex interplays through proteins, molecules, physical characteristics. In a way, we still don't fully understand to affect the disease risk, prognosis, and the recurrence as well. So I think epidemiology now is really exciting at the forefront of modern development of precision medicine or personal medicine. I mean, especially the study collect a large number of samples, different type of samples from a large number of individuals can, generate a lot of data, and then you can do various GWAS, PWAS, etc. Not only to inform understanding of disease etiology, but also to identify, certain sort of functional variants that can inform drug development or repurposing as well. And also informs the basic science research as well, animal experiments. So that's actually really exciting. So just to remind us some key strengths of a prospective studies as opposed to case control study in a surveys done pre-disease onset. So that's a little reverse causality. It doesn't really matter for genetic factor because it's a fixed, but it really matters for lifestyle factors, for biochemistry, etc. And also the control from same population, so less likely to be biased. And then if you have a stored biological samples, DNA, plasma, urine, fecal, saliva, which will facilitate hypothesis-free testing through high throughput omic assays. And then in the study, if you follow up enough long, you will accumulate a large number of disease events. This can greatly improve the sample size, but also the range of disease that can be investigated as well. But I think, the key strategic consideration for larger cohort study, I think there's several key elements. First thing is the size, because, when do not want to study just a few thousand, several, so want to collect a few or several thousand outcomes for specific disease. That's actually quite important. We also need heterogeneity, diversity. Not people overweight, obese, but also healthy, lean. So you can study a whole range of distribution. Same is true for adiposity, for the lipids. And also the exposure disease outcomes should be very well characterized, not just based on questionnaire, but also a wide range of assessment, maybe even using sort of state-of-the-art technology, wearable devices, so that you can actually characterize exposure or disease much more reliably. And then, because the prospect study is looking to the future, so it's really important you'll be able to follow up participants, not only their exposures in the changes over time, but also more importantly, disease outcome. If you cannot do that, it will become a kind of a cross-sectional study with a limited value. So to do that, you also need to develop infrastructure's capacity to ensure the study can really, continue long-term in, a few decades rather than just a few years. I just want to highlight that with a few kind of examples, size matters. Many of you probably have seen this. This just compare the association blood pressure with IHD mortality by age. If you study just 5,000 individuals, overall, you see an upward trend between blood pressure and IHD mortality. But you may conclude at age 80 to 89, high blood pressure is potentially protective. Is that true? If we increase sample size by 10-fold and then become more focused, clear, and this kind of, atypical ratios appear to disappear. But still, in young age group, you still have a J-shaped. So if we increase sample size by another 10 to 500,000 with a few 10,000 events, and then suddenly everything becomes so clear. So there's a strong log linear association between blood pressure and risk, irrespective of your age, and there's no any evidence of a threshold. So this is based on the PSC study, which bring together 60 studies, 20th century kind of a cohort study, and to meta-analyze. So this is really generate really reliable evidence. So this is actually quite important for the 21st century to consider really big. Because the factor we're studying probably not blood pressure, smoking, you probably have a more modest, but still very important relevance for understanding disease etiology. I mean, another thing is disease diagnosis. Compared with a case control study, you start with the people with a disease. So you tend to have a very good disease categorization, disease subtype, etc. But with a cohort study, you start with the individuals no disease. So this is a very challenge to follow them up, and also to try to get their reliable disease diagnosis, not just kind of a yes, no, but also some subtype, because they tend to review very different disease associations with a certain specific exposures. So for example, this is a paper we published many years ago looking at BMI esophagus cancer. As you can see, there's a complete opposite association, one positive with adenocarcinoma, and one inverse with squamous cell carcinoma. Of course, we don't know whether it's causal, so we're still doing research using Mendelian randomization, try to tease apart. But at least in terms of observational findings, there's a complete opposite. So if your follow-up cannot differentiate, the subtype, or the preference is very similar, and then you may find, there's no association at all. So this is actually very important. So I think how to get the disease follow-up done. I mean, routinely people tend to do active versus passive. I think in larger cohort studies, I think the most appropriate, if possible, you should consider very actively in the design phase and try to follow them up passively. That's through the linkage with the established registry hospital record, because they are much more cost-effective, very efficient, and the disease diagnosis tend to be much more reliable, and the data collection can be timely, more regular. So coverage tend to be very complete and less bias with incomplete coverage. But of course, there's a few caveats. You have to go through various approval procedure. You have to have a proper consent, etc. Of course, sometimes people do consider active follow-up. There's some advantages. But overall, this can be considered as a complementary strategy to passive follow-up. But otherwise, it's going to be very, very challenging. If you want to consider study last for, a few decades, you want to collect all the information, it's going to be very, very challenging. So, when you design study, you should consider very, very serious these issues rather than leave to the, after completion of recruitment. So another need is actually to study diverse population. Last year, Rory, myself, Empson wrote a commentary in the JAMA, argue for more studies in a diverse population. I think for obvious reasons, you should cover a much wide range of exposure, and a different population have a different genetic heterogeneity that can facilitate investigation. For example, East Asian fracture genes in the alcohol that can actually facilitate assessment of association moderate drinking with the cardiovascular disease. And the disease rate varies enormously. And for example, if you want to study stroke, the best place is not UK, not Japan, it's China, because China has one of the highest stroke rates now compared with other populations. And then different studies across different populations can also facilitate cross-validation, combined analysis, both for genetic and non-genetic. But overall, there's a substantial need to extend the study into different populations, which are not very well covered. For example, this shows the ancestry of GWAS participants still up to more recent years, still predominantly European population, with very little from East Asia, Southeast Asia, or African. We hope, over the coming years, the landscape will change, will improve significantly as well. So I just want to show one particular results to highlight the value of doing study in different populations. So this is a paper we published 2 years ago, Lancet, using the East Asia specific fracture genes to refute very reliably any beneficial effects of moderate drinking on cardiovascular disease. So in observational study, as in all the previous studies, as you can see, there's a J-shaped association between alcohol drinking and risk of incidence of ischemic stroke or hemorrhages. This is based on a very large number. And 95% actually confirmed by CT-MI. So we have very good disease outcome. So this is a moderate drinking, 1 to 2 units, 100 grams per week. It's about 1 to 2 units a day, has the lowest. But is this due to confounding bias, which is couldn't get rid of that? But in genetic studies, when we apply fracture genes, ADH, LDH, and to estimate the alcohol drinking, and as you see, there's no such sort of a J-shape or U-shape association, got a straight line. With moderate drinking, it's associated with 12 to 60% excess risk of a stroke. So this, I think this is actually quite important findings because for this lifestyle factor, you can't really do randomized controlled trials. You have to rely on genetic to some extent, tease apart causal effects. So I think the paper published and then summary conclusions, the genetic epidemiology shows that apparently protective effects of moderate alcohol drinking against stroke is largely non-causal. And alcohol consumption uniformly increased blood pressure and stroke risk, and appears in this one study of little effect on risk of myocardial infarction. Of course, with MI, we still want to get a bit more evidence so we can have a much clear, robust evidence on that as well. So I think this just highlights some of the background, line of thinking rationale for big core study, at least informs the way we design China caloric biobank studies. So China biobank study has been going on for over a decade, and it covers 10 geographically diverse areas. So 5 urban, 5 rural, from freezing north in Harbin to tropical source, from coast, well-developed urban cities to inland and rural areas. So with a very different disease rate, different stages of economic development, different lifestyle. So an overall, we have within 4 years recruited over 500,000 individuals with a lot of data collection by questionnaire and by physical measurement, most important with a collection of biological samples, blood, for long-term storage. So, and then subsequently we have done 3 re-surveys. Not everyone, that would be ideal, but it's just not feasible. So it's a random subset, 5%, 25,000, and repeat everything baseline, but with a lot of new enhancement, for example, ECG, carotid ultrasound, which were not possible baseline, almost 20 years ago, with the liver fibrosis, and now with eye test, with urine, saliva, fecal sample collection in the most recent ones. And we have a stored sample for nearly almost every individual. Now we're just after 15 years, now time has come for us to do large scale, high throughput omic, which require tiny amount of blood, 10 microliter, 100 microliter. You can generate maybe 100 or even 1,000 novel biomass, proteins, metabolites as well. So this is, over the next few years, we plan to do a lot more. And all the participants are being followed up indefinitely. So during the first 12 years follow-up, and we have recorded over 50,000 deaths, cause specific, over 1 million episodes of hospitalization of almost all the human conditions, 5,000, and with less than 1% loss to follow-up. So this is actually a highlight, initial some area selections and the strategies, system put in place. But this is still relatively early stage in terms of research because the data still increase quite rapidly through wearable devices, through linkage with other data set like NASA satellite for air pollution, but most importantly through large scale assays like whole genome sequence, proteomics, metabolomics as well. So I think this is really exciting. So in terms of disease outcome, it will capture almost everything. So they just give you a flavor about 25 different conditions. Some are very common, like ischemic stroke. Over 50,000 individuals develop ischemic stroke. Diabetes, almost 50,000. And cancer, COPD, fracture, cataract, to osteoporosis, retinopathy, schizophrenia, depression, dementia, snake bite as well in some rural areas, and earthquake. That's one area struck by earthquake, 2008. About a few dozen people died in 1 or 2 villages as well. So this just, demonstrate, we're able to actually link to the existing health insurance records to capture all the disease, a major disease outcome lead to the hospitalization. But I think, for several major diseases, we also undertake very careful outcome adjudication, try to validate diagnosis, but more importantly, try to subphenotype. Not only stroke, not only ischemic stroke, but actually whether it's large vessel, small vessel, cardioembolic, lacuna, asymptomatic or non-symptomatic, etc. For hemorrhagic stroke, whether it's lobar, non-lobar as well. So this is still ongoing process. So far, we have done about 80,000 through this kind of device we call PVD. Go back to original medical record and then have a group of specialists to review subphenotype as well. I think, over the last, 15 years, we have accumulated a lot of data. But until about 5 years ago, we're now to start to generate a lot of interesting scientific finding, because we have to actually have 10 years for study to mature, especially for the disease outcome to emerge. So now we have a lot of ongoing research activities. We have published 300 papers or submitted, plus over 100 active projects. We also have made data available to various researchers. So this involves a wide range of research areas, different disease, prospective analysis, lifestyle factors, blood pressure, adiposity, and some sort of nested case control study of particular elements like chronic infection with the cancer, risk prediction using genetic, non-genetic, GWAS of disease traits, lifestyle factors, PHIWAS, to validate particularly functional variants for drug discovery validation, and the Mendelian randomization, as I showed, ARCOR, various other risk factors to assess their causal kind of relevance for particular disease. And also some new areas of research like environmental health, healthcare delivery, utilization as well. So I'm not going to go through because it's, we have a lot of papers on the website if you're interested. So I think this just gives you a bit of background, where we are with the study. And then I want to step back, looking back retrospectively, our thought process during the study design and planning, organization, and management. What sort of lessons we learned? What's the thought process? I think this is probably quite generic to many of you. You all run your own studies, but some may be unique. Depends on your study population, study design. I think the Kadori is not hypothesis driven. So it's actually a complete open study. Compared with the older studies, you have to have a hypothesis like study smoking and mortality, British dot studies, or Framingham tried to study the causes of emerging rising burden of cardiovascular disease. But for Kadori, what we set out, we don't know what we're going to end up, because we want to collect samples. We don't know 10 years down the line, 20 years, what would become possible. So this is a very open-ended with very broad objectives. So I think in a way, conventionally, science, driving your plan, budget, timeline, output. But I think in reality, it's never worked like that, at least for our project. So I think at the time, after 10 years, finally, we got opportunity to get maybe $8 million to set up a study. So this actually budget, what we can get, within, maybe we can set up study within 5 years to recruit. So I think, we start with in a reverse order. This is a budget timeline. We want to get $500,000, very ambitious, no power calculation, to be honest. We just discussed with Richard, with Rory said, what would be the study looks really impressive. We said, okay, let's get 500,000, that will be one of the biggest. Of course, you can do power calculation retrospectively, but actually to start with, we just said, let's do 500,000. And with these constraints, so how are we going to deliver the study? So there are a lot of us thought through process, and also balance act, because a lot of things come to play. So, for example, if you want to collect everything, very detailed dietary questionnaire, which may, take 1 hour, so the recruitment rate is going to decrease, and the cost will increase. If you want to collect a lot of samples, urine samples, plasma sample, 50 ml samples, and then the cost will escalate. Not only the sample collection, but also storage. That probably need a few million instantly. And that, for example, if you want to do assay immediately, and then it will make the organization much more complicated, and the cost will escalate as well. So I think there are a lot of issues you need to consider. At least for us, we make decision, we want to do 500,000, and then in terms of data collection, we want to have a questionnaire, a key physical measurement. We want to have a blood. The whole kind of clinical visit time is not half day. It's probably around 1.5 hours. That's probably maximum. And with that, we estimated kind of likely recruitment rate. And this is actually can fit into the budget. This is still give us a very well-defined study with a lot of data, with the potential to enhance in future. So I think this is actually, quite, in retrospect, quite interesting kind of thought process. I'm sure many of you have a similar experience. Ideally, you want to start with the scientific questions, object, and then try to get funding. But in reality, we tend to work in reverse. We start with the likely budget, timeline, and then try to see, given the constraints, how much we can achieve. I mean, another thing is actually in a study population, sampling method, and whether you want to go for random representative samples or class samples. I think for us, we felt very strongly there's no need or it's not feasible to go for random samples. And then we just go to 10 carefully select areas, urban, with a very diversified areas, and then try to recruit as many as possible. We don't pay much attention to the response rate. Our response rate is around 35%. We can achieve 50, 60%, but actually the recruitment rate is going to drop quite substantially because, there's a long tail, people kind of do not come as you would expect it. So actually, we made a lot of kind of practical considerations in order to achieve the ultimate goal, which is 500,000, 4 years recruitment within budget. So I think eventually, what we have achieved is, what we set out to do. So I think in the interest of time, I'm just going to, describe some of the practical kind of considerations, how we set up to do the study to ensure the quality, to improve the efficiency of study management, and what sort of lessons we have learned. So once we have got protocol, SOPs, and the system, the first thing we come is how to develop quality assurance framework. This is critical for studying this kind of industry scale and operate thousands of miles away, diverse, dispersed area. I mean, anything can go wrong, will go wrong. So you have to have a very robust system. So this is the kind of framework we developed, not just plan, system, but also training and the mentoring, but also monitoring, reporting, but also risk-based kind of quality assurance. This is just a few examples. For example, we train staff with the dual roles, just in case some off sick. So, the clinical can still operate normally. And we operate in rural areas. The first day there were power cut due to, sandstorm, but luckily we got power generate, diesel generator. So actually studying, the clinic still operate normally. We always have a spare device, a laptop, just in case some kind of a breakdown or have defects. And in terms of local storage, we also always leave 50% freezer space, just in case one freezer got problem. So sample will get stored. So And then for the sample shipment to National Court Center, we always, for the sample from same individual, we never ship them same go. We always split, just in case there's a delay with the train or flight, and then sample got damaged or sold. And the instant date backup and centralized supply, and also to ensure the quality, we have an automated calibration maintenance schedule. So I think the key things are to reduce the human factor to the minimum. So you don't need to remember. The computer, the system will manage everything, will remind you. So that's, I think, quite important. In terms of monitoring, we also do weekly monitoring, daily monitoring across centers, staff, try to see the performance. This is just two examples, standing height, about months, one particularly staff. And this is blood pressure. We start to notice very early on, there's actually some issues going on. And then we realize later on it's not quality issues. This is actually variation to the temperature. So as winter comes, the blood pressure goes up, comes down, goes up, comes down. So eventually this leads to several really interesting publications, document the nice relation between ambient temperature and the blood pressure as well. So in terms of field work, it's very, very carefully managed and piloted. Like production line, for example, we have 18 staff, they properly trained. Average time is 1.5 hours. Number of recruit per day is around 60. And then that's per site, 10 sites operate simultaneously. So per year, it's 14,000 per site, and each year is 140,000. So I think, we stick with that schedule, we achieve that schedule. I think, everything done electronically with no paper involved. They come, they register according to the invitation, they present their national ID, and then the national ID, the photo get scanned automatically, and then synchronized across all the computer. So when they move to the next station, they scan their study ID, and then their picture pop up, and then you can check their identities. So this is actually quite important to make sure there's no kind of mislinkage. So this is the different stations, and all the data record directly into the computer and synchronized. And then end of the day is kind of downloaded and transferred to study center and sent over to coordinate center as well. So I think, we don't use actually bring your own device. All the computer are supplied by us, probably sort of set up. So you can't really use to watch movies or, log to the internet. It's specifically for study purpose. So this is actually quite important to maintain the security and integrity of the data as well. And then, at the time, 20 years ago, we have a lot of discussion to use a paper or computer-based questionnaire. So eventually we go for the computer, laptop, and now it's a quite standard norm. But at the time, people still use the paper-based. There's advantage, disadvantages. But I think overall, laptop-based computer file is actually far more efficient, far more cost-effective, far more flexible. So for example, you can have a, build in a lot of checks. You can have a build in a lot of sub forms, mental health forms, etc. And the data can be made available immediately. You can facilitate quality control, and they can be released immediately for analysis, etc. But of course, you need a lot of support, especially early stage, development. But that's actually, it's fine, the initial cost. So overall, we did right decision at the time to go for electronic sort of laptop questionnaire. So when we got the data, it's very high quality, no missing value, almost no logic errors. So this just shows interface we used. So you have a live system which record data. You also have a training system to facilitate training, and also have a built-in lot of logic checks and also audio recording as well. So you can actually listen, especially earlier phase, to facilitate training, quality control, and try to minimize errors as well. So in terms of questionnaire design, I know you all have a very good experience. I'm not going to say anything, except we've done a few pilot study. try to assess feasibility, and also build in a lot of checks. So this is actually behind the screen, you can't see. So for every page of a questionnaire, you probably end up with like 3-4 pages of notes, defines the digits, actions, movement, logic checks, soft range, hard range, and the missing values, genuine missing values, you should allow for that, for example, with a hash key, etc. So this is actually when the data is coming, it's all cleaned, validated, and you don't need to worry. So this is quite important. So with that, it's not just IT issues. It's actually need a lot of input from epidemiology scientists as well develop. So apart from questionnaire, we also have a lot of measurements. This is actually again quite complex process to decide which one to do. Of course, first thing, scientific kind of a consideration, but on top of that, there are a lot of other practical considerations as well to consider: cost, time required, complexity, the training need, workflow, whether can support Chinese, because our interface is always Chinese. So I think some are not possible to have an API. So this is one of these quite important consideration, and the data format size as well. So I think this always end up with a lot of a discussion. Even you decide you want to have a ECG, but which models? So this is actually quite time consuming, but this is actually a very important process as well. So as I mentioned, all the device connected to the computer, when measurement is done, the data automatically goes to the database, so you don't need to worry. And also even for measurements such as blood pressure, we develop a kind of device, you don't need to write down. Through the Bluetooth, you can actually link to the computer, automatically record it into the computer. You don't need to hand, written down. So this is actually quite important to minimize transcription error. But also the system also have to manage the process. For example, you have to have a 60 seconds break between two measurements. So there's a clock, count down 6 seconds, and after that, you press button. And if you don't do that, computer will record, will monitor, whether you observe that kind of standard procedures as well. So this is actually make life very easy for everyone. And also same time ensure the quality. The next big thing is the blood. This is the most important part of the biobank studies. Of course, you can collect a wide range of assays, but CKB baseline, we can only afford one tube, EDT tube of blood. There's many ways to collect, to separate aliquot. So traditionally you just write down, which is not viable. If you end up with millions aliquots. So now, luckily, we have a barcode system, whether it's 1D or 2D. At the time, this is not kind of possible or too expensive. So for long-term storage, we go for 1D, which is actually pre-printed in the audit kind of according to our specification, supplied by company. And then you just scan them, link them. In terms of sample aliquoting, we go for menu, but of course, big biobank studies, maybe USA studies, modern days, you can go for robots. So this is a UK biobank. Of course, there's advantages, very disadvantages with the manual operation. This is what we do. It's cheap but slow. The quality may not be perfect, and also you require careful training as well. With the machine-based, it's expensive to set up, but very fast, efficient. But sometimes you will experience some problems, which could be disastrous in a way. For example, if you have a leaked seal of the automated pipetting, and then sample will get diluted. I mean, this has happened in some studies. So this is actually, you need to really test quite carefully, make sure, this kind of errors does not happen. Otherwise, could, compromise the quality or the subsequent assays as well. I think the sample is really critical. To manage that, you need a lot of a system to record right linkage, to trace the movement. So in CKB, we have developed about 10 separate systems for sample collection, processing, movement, storage as well. I just show a few kind of photos to give a flavor. So this is a sample collected, and every half day they put in the actual box and the ship, with ambulance, with a siren, they can go through the checkpoint quickly, beat the traffic kind of lights, and deliver to the local laboratory, normally half-hour, 1-hour drive, and then get centrifuged and sub-aliquoted and put in the freezer. So I think this is a kind of standard procedure, but I think to do that, you also have a compute system. So this is what we call sample alley. So this is, specify how many samples you got, and the computer will guide you to retrieve relevant boxes, maybe full from previous days. And then you can link, the study ID imprinted on the EDT to the crowbar ID, which is a complete different set of a tube, and then make permanent marriage. This is a critical, because otherwise down the line, all the data will be linked back, traced back to all the other data as well. And then you can put various nodes, whether it's empty, whether penalized, hemolyzed, or sample conditions as well. And then this system manages a shipment. This is the coordinate centrally, by us. In Oxford, we can send requests, or Beijing can send requests. We can also monitor, the sample storage with alarm, with a backup generate as well. So this just shows, if you want to send 4 boxes from same individual, and then you got warning. This is not allowed. So this is just a highlight system we established to ensure sample absolute safety. So I think over the 4 years, we have shipped millions samples. We haven't lost any single samples. We haven't got any samples sold or damaged during the transit process. So this is actually a demonstration of robustness of the staff as well as the system as well. And when sample arrives, they go to the storage facilities. We don't have this kind of robotic as the UK Biobank, so we have actually sample storage purposely built in China, in a -80 freezers, a liquid nitrogen tank, and the plasma sample, urine sample also come to Oxford. So they stored, and then they manage it carefully with our computer system. You know exactly where they are. So you can trace, you can do inquiry, you can retrieve them for future assays as well. So this is actually quite important. This is built on kind of a careful study design monitoring. So I think in terms of lessons we learned is actually IT system play a really key critical kind of role in managing the study reliably, cost effectively, and efficiently. So I think in terms of IT system, and I just show a few examples: data collection, sample management, but there's still many other elements: data management, assets management, standard monitoring, access control, which need a lot of computer support as well. So this is just specific system we developed. Give you some flavors. Each one just one specific system. Some are actually web-based, some are actually C++, etc., depending on the need where they operate. For example, I haven't had to show you some outcome adjudication. We call PVD. iCase with a disease kind of a subphenotyping. And this MMS, this is a system to manage supply, to manage calibration, to manage the maintenance. So there's automated kind of schedule. We send out remind automatically and depends the need. So you don't need to remember. So this is a research tracker to manage in our research project, and the link to study ID, link to the data delivered, and the publication as well. So this just shows the diagram, how we operate. This is Oxford. We have a national center in China. We have 10 sites. We also have some strategic partners like BGI. So all connected somehow through computers, some various form of a system we build. So it's kind of an ecosystem. They operate quite efficiently. So we're still developing new things because there's a new need as well. So I think in the last few slides, in terms of IT development, I'm not expert, but I learned quite a lot through the process. I think what would define a good system, security, compatibility, flexibility, ease of use, efficiency, reliability. I mean, this is common to all the system. But I think what we have done is follow the best practice in the IT industry. So in terms of a development, this kind of work for process. This is a simple version. Firstly, you need to analyze the need, and then develop a plan, and then write URS, user requirement specification. So this is actually very important. This does not come from programmers. This comes from researchers, from epidemiologists, from field work. They specify a requirement, and then program the right code, and then formal testing, and then revise, and then develop user and deploy, and then this is kind of cycle. You may detect some bugs, you want to improve it as well. So this is actually standard process for every single ones. Of course, there's various ways developing software. For example, outsource, you can get company to do for you. You can also develop in-house. So for us, everything done in-house. And although initial cost is high, time delay, but the return is very substantial. The easiness with maintenance, upgrading, quality control, integration, and the overall cost is relatively low compared with outsource. So I think, in retrospect, I think we made a right decision to do that as well. And of course, there are a lot of issues with infrastructure, hardware. It just shows very sophisticated arrangement, regional study centers, national centers. So overall, we have 30 servers across 3 locations, and 120 desktop, 40 tablet computers for outcome adjudication, and over 100 laptops, at one particular time, for field work as well. So it's quite big, sort of challenging to maintain integrity. So last few slides just on the data. I'm not going to go into the details in the interest of time. So when data arrives, they're cleaned, but there's still quite a lot of work to do. Try to process, try to link them, try to make them available, and try to de-identify, remove sensitive information like study ID. Try to generate, so de-identified analysis database for internal use and for collaborators as well. So with that, we also have established kind of core management approaches. There are a few principles, I think, which apply not to our study or other data management, privacy, security, restricted access, anonymized data for analysis, encryption, for data delivery, and also careful documentation, all the process, IOPs, variables, etc. That provide simple data request delivery, so it's relatively straightforward. I don't know, we also make it available to academic researchers on the website, so you probably have some of you may have some experience as well. So very last slide is actually where we are. It's still relatively early stage. The next 5-10 years it's going to be really exciting, because now we want to turn stored sample into the data. Like omics, metabolomics, proteomics, whole genome sequence. So across all this, we have done a few pilot studies, really exciting. They don't, it's still relatively expensive, but the price is coming down, like whole genome sequence, $200 maybe. So our immediate plan is try to move into the cohort 1, at least to get the 100,000 done next few years, and then get whole cohort done, and then make data more widely available to the research communities. So this will greatly improve our understanding biology, drug target discovery, detection, diagnosis. But with that, it will come with a lot of challenges, and for data management, and so we need to think, novel ways managing, analyzing data as well. So I'm going to stop here. Recently, we just published a book, summarize some of our experience. So if you're interested, you can get hold of that Amazon, or I'm quite happy to send you a PDF copy if you want. Yeah, I'll stop here. Thanks very much for your attention.
Yeah, thank you so much, Zhengming. And congratulations not only on the book, but on what you've been able to do with the China Kadoorie Biobank. And a number of comments in the chat are also giving you congratulations. We have time for some questions. There are already a couple that have been asked. One from Albert Tanisa, I hope I'm pronouncing his name correctly, who I believe is from Scotland, asking what the percentage of blood and non-blood relatives accidentally are recruiting into CKB.
Over about 20%, they are related. Some are kind of first degree, some kind of a second degree. So yeah.
Related to that though, what is your, are there return results policies that you have? And if people, how do you deal with just maybe non-paternity or paternity issues that you might find in the course of your studies?
So I think in our written informed consent, we made it very clear the return results. So certainly anything happened on the day, we provide a detailed leaflet with like health information and their results. Not only kind of questionnaire, smoking, drinking, some kind of health advice, whether they make difference or not, we don't know. But like adiposity, blood pressure, lung function, as well as some spot blood tests like lipids, the blood glucose as well, HB, surface antigen. So we did do a few tests, and which is available for all half-million. So that results actually given immediately. But with other test results, we said we're not going to return because we're not going to match them immediately. So they understand. So overall, the sample collection is very high. Only less than 100 samples are not collected out of 500,000. So almost 100%.
Thank you. We have a question from Bruna Galabardes from the Wellcome Trust about how many iterative cycles did you go through before you felt like you had the final process in place? I think this is related to some of the IT cycles that you showed us.
You mean the pilot testing of the system? Is that IT system or?
Bruna, do you want to elaborate if you can unmute?
Yes, exactly. That was when you were explaining that. I just wondered, did you have the vision of the process in place already, or how long did it take, and did you have to change?
Yeah, I think there are a lot of test dry runs. I mean, first we develop system and then pilot tested. Some actually go to rural settings to have a small recruitment. We don't record the data. I mean, we don't use the data, try to get feedback. And then try to iron out all the issues, translation issue, language issues, etc. Another thing is actually, and trying to bring together the whole system need a lot of testing. So we have a lot of dry runs in Oxford, 3, 4, 5, and then move to China and then move to clinics as well. So I think, really depends. It's, you do need to have a lot of piloting. So that's actually paid off quite significantly as well. Of course, even you do that, things can go wrong, will go wrong. And then the good things, we can actually send out the patches very quickly, almost within the next day. Sometimes they report some issues. So we have IT standby. They can identify, they can fix it within a day, and then the new version deployment will be on the way, immediately. So yeah.
Thank you. I want to continue with we have a few more questions. Hopefully we'll get to all of them. One is, can you tell from Caterina Ferriccio from Chile about the sharing data and samples with third parties? Can you describe the system for sharing data and samples with third parties?
Yeah, I think if you log into our website, we have of course, the website, and now we're just undergoing, very substantial revision, new system. But existing ones, there's a section on the data sharing and access, so anyone can apply, but not the blood samples because we only have limited samples. So what we want to do is actually centralize, and then come up with a plan to maximize the potential, do cohort-wide, not just specific questions, omic and generate a lot of data. And that data can be made available. So I think we have released all the baseline data, all the race survey, and also mortality, and the morbidity will be added. So, any researcher can apply.
Great. And I also should have mentioned that Katerina, who asked that question, is our speaker for June 24th. That information is also in the chat. So Hussein Pushi from Iran is asking about the funding strategy here. Is this like one big project, or are you now funding it as a series of smaller projects?
I think to start with, is one-off funding from Kadoorie. Well, that's why it's called China Kadoorie. I didn't explain. So this is a charitable organization in Hong Kong. We approached, and then they give us a one-off funding. The only condition is bear the name, the study bear the name Kadoorie. That's it. So that's actually 7 million. We also have some core funding, maybe 1 or 2 million. So total budget to set studies, let's say, like 8 million at the time. And then once we set up study, and Wellcome has very generally support study, and then a few MRC, CRUK, and the China as well. But I think to start with, it's very challenging. It took us 10 years to get funding because at the time it was a bit too ambitious, too vague, whether it's possible, what's scientific value. But and subsequently, we got very generous support. Yeah.
Thank you. Michelle Ramsey from Africa is asking about the consent model that you used. How long does it take you to consent individuals, for all the aspects of data sharing, future projects, future use of data, future use of samples?
I think for the consent, what we did is, I think potentially relevant is to not do just consent in the clinic. That's actually, you waste a lot of precious time. So actually in China, we're quite fortunate to have a very detailed participant list we get hold in advance. So before we go to village, we know exactly how many people. So we can actually distribute some leaflet, explain study, and also have some social mobilization, discuss with the community leaders, and explain, have a kind of, PPI kind of a group meetings. And then we send out invitation and encourage them to come. When they come, we answer questions, explain again. By then, most of them quite consent. If they don't consent, they don't come. That's why we do not get 100%, but that's fine. We explain, why we do this, what we involve. So the overall response rate, as I said, it's around 35. Some are maybe a bit high, yeah.
Thank you. And I'd just like to ask one more question, perhaps at the risk of going a few seconds over time. But you mentioned that you published 300 papers and you're from a Department of Population Health at Oxford. How do you measure the population health impact of the biobank?
It's good questions. I think this first thing got to be long term. I think, if you look back, some of our results have a quite immediate impact. Our report on smoking has informed the policy change in China, tobacco control. Our alcohol results certainly, has informed, some policy in a number of countries. Even before our publication, people chasing me, can share the results because they're developing some policies, etc. So certainly, has a feed into policy changes. Another thing's kind of genetic studies, really important. Some drug, development, we're developing FIWAS, CTP, various things. The results are highly consistent. I think this has also informed the drug company development strategies. Before you launch expensive trial, you need to consider genetic evidence. They don't cost money. So now we have some good collaboration with a number of farmers, whether it's kind of specific small project test one specific functional variants present only in Chinese or not. So I think, I think another key things with the founder with this is actually out management. For the first 10 years, you should set different output. It's not scientific output, it's actually get study set up. So that's actually different. And very often you try to mix two things. You try to do recruitment as well as a publication, and then you can't do both. I think that's actually quite important. Unless like a nationwide effort, like all of us, you can do. But for us, all our effort, for the first 10 years, no publication, almost. Mm-hmm. I mean, and then from 2015, suddenly, got rapid increase as well.
Well, thank you, Zhengming. Congratulations again. Also, we are grateful for you to be our inaugural speaker for this IHCC educational seminar series. Really appreciate also the attendees from all over the world that joined us today, and welcome you back on June 24th at 12 o'clock UTC for Caterina Federecchio from Chile, who will be talking about the Mako population-based cohort. So with that, I'll wish everybody a great weekend and good health and safety.
Thank you. Thank you.
And recording will be available.
Yeah.
On our website.
Yeah.
Thanks, Zhengming.
Thank you for sharing. Yeah. Bye.
Thank you,
Bye-bye.
everyone.
Bye.